Hello, my name is Emily Halphen, and I will be presenting transplantable pancreatic islet organoids derived from feline reproductive organ-associated adipose multipotent stromal cells. Diabetes mellitus is a growing threat to cats, affecting between 0.2 and 1% of client-owned populations. Like human diabetes, the majority of cases are type 2, which is characterized by initial insulin resistance and ultimate beta cell failure. Current therapies are palliative with lifelong insulin supplementation, which can be cost prohibitive. Feline adipose multipotent stromal cells from reproductive organs, which are medical waste of elective surgeries, have been reported to have robust proliferation and multipotential. FASCs, when differentiated into insulin-producing beta cells, may provide a permanent cure for patients with type 2 diabetes. Based on previous beta cell differentiation with human adipose tissue, we designed staged induction protocol to differentiate FASCs towards beta-like cells. We hypothesized that beta-like cells prepared with our novel protocol would upregulate pancreatic cell-associated gene expressions, synthesize insulin, respond to glucose, and effectively correct hyperglycemia in diabetic mice. To test this hypothesis, our objective was to quantify the pancreatic cell-associated gene expressions, detect synthesized intracellular insulin, quantify insulin released upon glucose stimulus, and evaluate the glycemic levels in diabetic mice transplanted with the differentiated beta-like cells. First, FASCs were isolated from epididymal and broad ligament adipose tissue obtained from elective surgeries and expanded to passage 3. The FASCs were then differentiated towards insulin-producing beta-like cells in a 3-stage induction protocol with defined culture mediums for each stage, which formed pancreatic organoids, as seen in Figure 1. Data was obtained by evaluating the formed pancreatic organoids for viability with calcium staining, cell-associated gene expression with quantitative PCR, insulin release upon glucose stimulation with ELISA, proteomes with mass spectrometry, and intracellular pancreatic hormone synthesis with immunocytochemistry, immunohistochemistry, and immunotransmission electron microscopy. Additionally, organoids equivalent to 10 million cells were transplanted intraperitoneally to athymic diabetic mice for correction of hyperglycemia. Figure 2 shows the calcium staining of some of our cells to test the viability. The majority of the FASCs were alive, which is shown with the green color, and fewer cells were dead, which is depicted with a red color. Figure 3 shows the evaluation of gene expression levels of pancreatic cell-associated transcription factors. Beta-like stem cell markers were upregulated incrementally with advancing induction stages. Notably, the highest upregulation was observed at stage 3. And expression level differences between male and female donors existed for the nestin and neurod1 markers, with higher expression in cells from female donors. Figure 4 shows that the pancreatic hormonal genes for glucagon, insulin, and somatostatin were upregulated incrementally with the highest induction at stage 3. Upregulation of the insulin gene was approximately 10-fold higher than the other hormones, which indicated differentiated cells' genetic profiles to be closer to beta cells than alpha or gamma cells. Figure 5 shows the evaluation of organoid responsiveness to glucose. Organoids were stimulated with low and high concentrations of glucose repeatedly for 30 minutes, with the exception of the first stimulus, which was a low concentration for 2 hours, and the last stimulus, which was a depolarization of cells with potassium chloride. Although statistical differences were observed, feline pancreatic organoids responded to serial glucose stimulus in an apparent dose-dependent manner, which implies their functionality as beta cells. Figure 6 shows the evaluation of the comprehensive profiles of the pancreatic organoids' proteomes. The Venn diagram compares our experimental FASCs to a control sample proliferated in identical medium. This diagram shows that 180 proteins were upregulated and 425 proteins were downregulated in the pancreatic organoids in comparison to the control. The scatter plot shows the differentiated cells had different expression levels than the control, with notable upregulated proteins including glypican 1 and 6, which are markers for pancreatic progenitor cells. Figure 7 shows the presence of pancreatic hormones which were found through immunocytochemistry staining of undifferentiated clusters in pancreatic organoids. Clusters were labeled with insulin in green and somatostatin as red. Cell nuclei were counterstained with DAPI and are shown in blue. Abundant insulin was observed in pancreatic differentiated clusters, while somatostatin was absent. Figure 8 shows immunocytochemistry staining on the single-cell level to evaluate the synthesis of pancreatic hormones. The top panels are a control of feline pancreatic islets to demonstrate the natural structure cells will form within these islets. Feline islet cells and pancreatic organoids are labeled with insulin in green, glucagon and somatostatin in red, and the nucleus counterstained with DAPI in blue. It was observed that the feline islets mostly consisted of insulin-positive beta cells, glucagon-positive alpha cells, and a few somatostatin-positive gamma cells. On the other hand, the pancreatic organoids consisted solely of insulin-positive beta-like cells. Figure 9 shows that the predominant presence of intracellular insulin was also confirmed ultrastructurally. When the ultrastructure of the pancreatic organoids was examined with transmission electron microscopy, the cells within the pancreatic organoids were shown to have formed numerous vacuoles, indicating differentiation towards secretory cells. Further, with immunogold staining, insulin was shown to be the predominant hormone within the cells. Insulin was indicated with white arrows, glucagon was indicated with black arrows, and somatostatin was absent. Figure 10 shows the in vivo evaluation of functionality in the pancreatic organoids when injected into immunocompromised athymic diabetic mice. Each mouse received a total of 10 million cell clusters in an intraperitoneal injection. Fasting glucose levels were recorded every morning for 10 days, 2 of which were prior to the injection. The graph shows an immediate correction of hyperglycemia within 1 day of treatment. The correction lasted until day 3 post-treatment and relapsed by day 5. Our studies have indicated that FASCs were able to differentiate towards beta-like cells with single hormonal synthesis of insulin. However, simultaneous expression of glucagon and somatostatin genes at lower levels, combined with relatively weak insulin release responses upon serial glucose stimulation, have suggested that further maturation is necessary, which may ultimately achieve long-term correction of hypoglycemia in diabetic animals. Collectively, the pancreatic organoids derived from FASCs with our novel differentiation protocol have the potential to cure insulin-deficient diabetic patients in feline cases and beyond.